but a lot of hype was created with this discovery that not all messenger RNAs are being translated into proteins, but that messenger RNAs can be dragged away from the ribosome into the RNA-induced silencing complex, where messenger RNAs then are being repressed or even degraded. And this competition between ribosomes and these complexes, these RISC complexes, is being mediated by these microRNAs, non-coding, short non-coding RNAs, microRNAs, or there are other types of non-coding RNAs. Their job is to drag away a messenger RNA into the RISC complex for repression of translation. So this is an endogenous mechanism of regulation of gene expression, which turns out to be very important during cellular differentiation, during cell division, and also in many, many diseases. There are numerous studies now showing that dysregulation of microRNAs is associated and maybe even the cause of certain diseases. So the microRNAs turn out to be as key regulators, as key switches in the regulation of cellular differentiation, and also plays therefore an important role in diseases, and are new drug targets, not only tools but new drug targets for RNAi-mediated therapeutic modulation. And without going into detail, there are now different ways of modulating this microRNA maturation in the cell. This is a stepwise process. Pre-microRNAs are being matured into the microRNAs that then repress translation of the target messenger RNA. And this can be modulated either by overexpression of microRNAs, by viral delivery, or by synthetic microRNAs, which are being introduced into the cell. These are called microRNA mimics. Or you can inhibit the function of microRNAs with antisense oligonucleotides called antimirs, and thereby modulate gene expression in general and gene expression in certain disease states. So these are targets, but at the same time the oligonucleotides are the tools. And in a very similar mechanism, this can be also exploited for any target gene, not only microRNA targeted genes, but any gene in the genome, by using synthetic siRNAs, or short interfering RNAs, which can act on the same pathway, but then modulate any target gene that you can select out of the genome. And you can actually deliver those via viruses or directly as synthetic siRNAs, and that's clearly the goal. And here again, we have then the problem of delivery, because oligonucleotides are large molecules, larger than peptides. They have in the range of, so double-stranded RNAs are in the range of 14 to 16 kilodalton, and they are not being taken up by the cell that easily and do not penetrate biological barriers. So this was actually here indicating there are a lot of problems with this approach. There's a lot of hype, but there are also a lot of problems, and the key problem is delivery. So what we really have, this was a snapshot of new biologic-based therapeutic modalities, and but all these modalities have one problem in common, and that's delivery. They are too big. Biological barriers actually are designed, and natural design is to prevent uptake of large molecules. This is a defense mechanism against viruses, against bacteria, against toxins. And by bringing these molecules into the cell, one has actually to bypass this normal barrier, this normal defense mechanism. So there's limited tissue accessibility of all these molecules, limited delivery across the biological barriers, therefore difficult, therefore oral uptake is very difficult, if not impossible. There is poor delivery across cellular membranes, therefore therapeutic antibodies are currently limited to extracellular targets, and actually we are running out of those targets. That the easy targets have been selected already. If the ideal sort of future of therapeutic antibodies is to bring therapeutic antibodies or the smaller fragments or scaffolds into the cell, so that these antibodies or non-antibody scaffolds can target intracellular targets. So that's one desire of this, or one objectives of this consortium, as we will just summarize. So there are undesirable pharmacokinetic properties of these molecules. So some have a short half-life, like oligonucleotides. They are rapidly excreted. Large molecules are quickly filtered out by the kidney or taken up by the reticuloendothelial system. There's reduced stability. And these problems need to be addressed for successful delivery. There's also the potential for immunogenicity, which can be partly prevented by choosing the right nanocarriers. And another problem for the industry is that poor delivery results in can result in high cost of goods, because you have to apply a lot of these molecules in order to get a therapeutic effect. So the objectives, therefore, of this consortium are to improve the basic understanding of the intracellular uptake mechanisms for these molecules, for these biologics, biomacromolecular drugs, proteins, peptides, oligonucleotides, because only if we understand the basic mechanism of crossing biological barriers and of intracellular uptake, only if we understand this, then we can actually develop and employ strategies for successful delivery. So the second key objectives is then to use this know-how, this knowledge, to develop nanocarriers that succeed in delivering these drugs across the epithelial barriers. And here are examples. One key example is certainly intestinal barrier for oral uptake, and nanocarriers that succeed in bringing these molecules into the cells. So cross the cellular membranes. And as I pointed out, intestinal barrier, crossing the intestinal barrier is required for successful oral uptake of these drugs. And that's going to be the holy grail of biologic-based therapeutics, to deliver these molecules orally, just like a normal small molecule pill. And this, to achieve these objectives, this is certainly a cross-functional, that is the requirement for a cross-functional approach for a consortium, basically between public and private organizations, because we in the pharma companies, we actually do not have the resources and the time to do the fundamental research. That's going to be done or is being done in academia or in specialized companies like biotech companies. It requires tangential research. A lot of disciplines are involved or need to be involved, like chemistry, molecular biology, cell biology, nanotechnology. And so this is a typical, or clearly a typical topic for a consortium like IMI, with cross-functional and cross-institutional contributions from various experts from academia, small, medium enterprises, biotech companies, regulatory and industrial scientists.